In this video, I am going to talk about PCR or polymerase chain reaction. It is a technique used in molecular biology experiments for the amplification of DNA fragments. What does DNA amplification mean? Suppose you have one strand of DNA and you want to make copies of it. PCR helps you make several copies of that DNA, usually millions of copies, in a very short span of time. That is the main advantage of using PCR. The technique was invented by Carey Mullis in the year 1983. Before going into the process of PCR, let me explain the components of a PCR reaction mixture. In a PCR tube, the following components are added in the given order. At first, sterile water is added. In that, assay buffer is added, which serves to give a stable chemical environment for the functioning of DNA polymerase. Then the DNTP mix is added, which contains adenine, thymine, guanine and cytosine. After that, the forward and the reverse primers are added. Then the template DNA is added, which serves as the source from which the copies are to be made. And finally, the DNA polymerase is added. The reason for adding DNA polymerase at last is that if you add it first and then add sterile water or buffer in it, it can damage the DNA polymerase as it is very fragile. Therefore, adding it at last, the sterile water and buffer acts as a cushion and therefore helps to protect it. The PCR is generally carried out at 94 degrees Celsius, at which the DNA polymerase can melt. Therefore, instead of normal DNA polymerase from the bacteria Thermus aquaticus is used. It is called tagged DNA polymerase. Now let's look at the process of a PCR reaction. Suppose this is a double-stranded DNA. When we apply heat to the double-stranded DNA, the hydrogen bond between the two strands, as they are very weak, they break down. Now that there are no chemical bonds between the two strands, they separate into two separate strands and form single-stranded DNA. This process of formation of single-stranded DNA from double-stranded DNA is called denaturation. Generally, in a PCR reaction, this process takes place at 94 degrees Celsius.
Let me draw the single-stranded DNA here once again. The primers that we said before, the forward and the reverse primers, now join with the single-stranded DNA. This will be the 5' and 3' ends of the single-stranded DNA and the primers.
Now, tagged DNA polymerase attaches at the site where the primers join with the single-stranded DNA, and then it starts the process of elongation. After both the strands of the DNA are completely copied, we get two double-stranded DNA. Each double-stranded DNA has one new strand and one old strand. Here we can see that at the start there is one double-stranded DNA and at the end there are two double-stranded DNA, and this completes one cycle of a PCR reaction. When we apply heat to these two new double-stranded DNA, 4 single-stranded DNA forms. Again the primer and DNA polymerase joins and the elongation happens, and this process continues. The process of joining of primers is called primer annealing, and it happens at 48 degrees Celsius, while the process of elongation happens at 72 degrees Celsius. Here we can see that at the end of the second cycle there are 4 double-stranded DNA. The total number of DNA after a reaction can be calculated by the formula 2 to the power n, where n is the number of cycles. Suppose there are 30 cycles. That means after the end of 30 cycles there will be approximately more than a billion DNA molecules present in the reaction mixture.
The PCR reaction takes place in a machine called a thermocycler. At the front of it, there are buttons which enables us to program the temperature according to our choice. At the top of it, there are small chambers in which we put our PCR tube. Then the machine provides the temperature for denaturation, annealing, and extension.
If we look at the graph of a PCR reaction, it looks something like this. The y-axis denotes the temperature while the x-axis denotes the time. The first peak is that of denaturation, the second of primer annealing, and the third of extension. So here 94 degrees Celsius for denaturation, 48 degrees Celsius for annealing, and 72 for extension, and the rest for the following cycles. From here to here, it represents one cycle.